Hello, my name is Graeme Oatley, and I'm a senior research assistant working on the Aquatic Symbiosis Genomics project in the Tree of Life core laboratory. Today, I'll be talking about how our work in the core lab goes towards generating high-quality genomes and discuss the challenges we face dealing with ASG samples, but also highlight some successes we have achieved. The aim of the Aquatic Symbiosis or ASG project is to sequence the genomes of 500 symbiotic relationships. This roughly translates to sequencing the genomes of 1,000 species. Samples for the project have been selected and collected by 10 global hubs. Each hub shares a biological focus in terms of taxonomy, ecosystem, or mode of association. These 10 hubs are taxonomically diverse and cover both marine and freshwater ecosystems. Here I provide a summary slide of the diversity of the ASG samples that have been and we expect to see coming through the Tree of Life core lab. As you can see, samples range from projects investigating coral and sponge symbioses, marine lichens, and annelid chemosymbiosis, just to name a few. Once samples are collected, they are sent to the Sanger Institute and enter our genome engine pipeline. This schematic is often used in the Tree of Life and ASG talks, and neatly shows the pathway from sample collection to the final product of a genome in the form of data on the Open Data portal and publication of genome notes. Once samples have been collected, identified, and shipped to the Sanger Institute, they are handed over to the Tree of Life core lab, shown here in the red box. Once in our freezers, samples are prepared for high-C sequencing, which is often performed in another lab at Sanger, and for DNA and RNA extraction within the core lab. Post-inspection of the samples, they are divided into those that will be disrupted using a power masher, which is a handheld motorized pestle and mortar, or a Covaris cryoprep, an instrument which utilizes high impacts to disrupt frozen material into fine powder. For DNA extraction, we routinely use the bead-based QIAGEN MagAttract extraction kit to obtain high molecular weight DNA. Post-extraction, we assess DNA yield on the qubit, DNA quality on the femtopulse. If sufficient yield and quality is obtained, DNA is prepared for PacBio sequencing in our labs before being submitted for sequencing at another team at Sanger. Similarly, for RNA, we utilize a bead-based extraction kit and assess quality and purity using the qubit, nanodrop, and bioanalyzer before submitting for RNA sequencing. To date, for ASG, we have performed 439 DNA extractions from 176 species. All species have had material submitted for high-C, although very little high-C data are available for corals and sponges, as protocols are still being developed for these sample types. RNA has been a very recent addition to the core lab pipeline, as approximately 40 ASG species have had RNA extractions, 36 of which have been submitted for sequencing. As you can imagine, ASG samples have provided some challenges when it comes to extracting sufficient high molecular weight DNA required for sequencing. I will briefly touch on some of these challenges, but also mention successes that we have achieved in the lab so far. Specifically, I'll be looking at three groups: the Cnidaria, which are your corals and jellyfish, Porifera, which are your sponges, as well as the Mollusca, which are your clams and squids and the like. First, let me start with the Cnidaria, and specifically the corals. The majority of coral samples have a calcium carbonate skeleton, which is very hard, and breaking through the skeleton to get access to the coral polyps is very difficult. We have been utilizing the cryoprep to disrupt frozen coral samples, but have found that coral samples often damage the internal impact plate of the instrument. A damaged impact plate results in holes forming in the cryoprep bags. So these are bags where we put the samples in during the disruption process, and they can result in the loss of samples during the disruption process, something we are keen to avoid. Therefore, figuring out the best way to disrupt corals is currently an issue we are having to deal with in the lab. As the majority of samples is made up of skeleton, a second issue with corals has been determining the amount of material to put forward for RNA and DNA extraction. For DNA extractions, while our initial work suggests that about 100 milligrams of starting material should yield sufficient DNA, this has not been consistently shown across all coral species extracted so far. Given this, we are currently experiencing a 40% success rate for all Cnidarian DNA extractions. Next up are the sponges, another very tricky group to work on. We have often been able to recover very high DNA from sponges, However, the quality from many extractions was never good enough to proceed with sequencing. After sharing these results with our sponge colleagues, Joe Lopez suggested trying a squeeze method to try and improve DNA quality. Essentially, this method entails squeezing a sponge sample in a tris EDTA sodium chloride buffer to get sponge cells from the skeleton, and then using those cells with your extraction method of choice, in our case, the Magatrack kit. Here we have a femto profile of sponge extractions from Strypnis fortis, first using standard cryoprep samples. As you can see from that, the yield for both these extractions was good, over 4,000 nanograms. However, the DNA was mostly low molecular weight DNA. However, applying the squeeze method to the same species resulted in not only higher DNA yields, but also greatly improved the quality of the extracted DNA. As a second example, here are two extractions from Spongia officinalis using the cryoprep disrupted tissue with variable DNA yields, but both with very low molecular weight femto profiles. And while applying the squeeze method to the same species did improve the DNA yield, the quality of the extraction was not improved. While the use of the squeeze method has shown to be beneficial for obtaining high yield and high molecular weight DNA from some species, it doesn't appear to be a solution for all species. And finally, I'd like to finish off with the molluscs. To date, the molluscs have shown a very good DNA extraction success rate, with many species being submitted for PacBio sequencing. Additionally, metazoa high C protocols appear to work well for this group. However, unlike coral and sponges, molluscan symbionts are often found in specific parts of the host organism. This, ensuring that symbiont-containing tissues is selected for extraction, is very important. An initial assembly of the species of the giant clam, Tridacnocrophea, lacked any symbiont sequence data and caused some head scratching until it was realized that host-only tissue was selected for the initial DNA extraction. Symbiote-containing tissue was quickly extracted and submitted for sequencing to generate data to be added to the initial assembly. In summary, the ASG project has and continues to throw up numerous challenges to the Tree of Life core lab, and I expect the same to happen for the assembly and curation teams further down the pipeline. But that's what makes it an exciting project to be a part of. The team has made some very good progress at extracting DNA from species supplied from the ASG hubs, and we're very encouraged to see the sequence data that is being produced by the assembly team. And to add to that, I'm very excited to see what the hubs that have supplied the samples will do with that data once it is made available. And with that, I would like to thank you all for listening.